Good afternoon. Now we are halfway through last class. We will finish the lesson to the end. Yesterday, if you remember, we saw fertilization and what are the important points in an exam point of view. I gave you. So whichever part which you are doing, you have to focus on the multiple choice and the NEET question part, because mainly you are focusing on the NEET coaching more than for your completing the portion. Okay. So fertilization, when does it occur? Where does it occur? What is the importance of the nuclei? How does the haploid nuclei, diploid nuclei, zygote, zygospore, diplomatic cycle, haplomatic life cycle, all this you can. And during the fertilization, there's one thing will be given in your book, that is sex determination. Now you know where is sex determination, and in between a small paragraph. So after fertilization, the resultant cell in a human body, zygote, you know that. So the zygote, the sex determination of the zygote is mainly by the sex chromosome. Now that also I told you, sex chromosome is that is X and X and X and Y. Where is the sex chromosome? Now next that in between after fertilization, along the fertilization, end of the fertilization, it will be given in your textbook. Sex determination. Okay, sex determination. I have to go in order. 1 second, please. I will go over to the same. So, along with the fertilization, end of the fertilization, sex determination. Determination. Now, since it is referred, you already know in your genetics. Now, sex determination is by the sex hormone. I'm sorry, sex chromosome. Now, where is the sex chromosome? Which cell sex chromosome? Invariably, the children will have an idea sex chromosome is present only in the reproductive organ or the cells of the ovary or only in the gamete. That's a wrong notion, okay? Sex chromosome is present in each and every cell of your body. Now, there are 46 chromosomes, the 23 pair. Now, to know this 46 chromosomes, 46 is a diploid number in our body. 1 minute. 46 chromosome is a 2n, and in each and every cell 46 chromosome. This can be arranged in 23 pairs. Now, these 23 pairs are identical in male as well as in female. These 23 pairs are similar in male as well as in female, all the 23 pairs. Now, of this, not all the 22 in male and female. In female, if you take of this 23 pair, 22 pairs are identical in male and female, but each pair is different from one another. And you can group them according to the length of the chromosome into A, B, C, D, different groups. But you can easily find out which are longer, which are shorter chromosomes, and the chromosomes can be, the 23 pairs can be put up into different groups. The grouping up of the 23 pair according to the structure of the chromosome. What is meant by structure of the chromosome? Centromere and the length of the arm. So the 23 pairs can be grouped into different groups. Of this, the two groups are 22 pairs of 22 groups are identical in male and female, and the 23rd pair. The 23rd pair, which is dissimilar in the male and female, is known as the sex chromosome. Now, this sex chromosome, as you know, all the chromosomes are in pairs, diploid number, okay? And the sex chromosome in the female is denoted as X and X. In the male, it is denoted as X and Y. And X and X, in a sense, the chromosomes of two chromosomes are identical. That is, if you take X chromosome, this is the same. These two chromosomes are identical. You call them as X and X. In the case of female, one chromosome is longer, you call that as a X, and the other one is shorter, you denote it as Y. But the X chromosome of the female and X chromosome of the male are identical. So the female and male resemble 50% each other. So genetically, no female is a perfect female and no male is a perfect male. And another thing, the DNA content is 25%. They have got lesser in male than in the female. Now, these sex chromosomes separate during gametic formation. So if you take the sex chromosomes X and X in the case of female, and in the case of male X and Y, now they segregate into X and Y in the gamete by meiosis. Meiosis or reduction division. Similarly, these are the gametes. Now these are the female gamete, the male gamete. So this can combine with this, it can combine with this. So the male gamete, when it comes, it will become, the product will be X and X or X and Y. Similarly, this thing, if we take, the product is X and X or X and Y. So that zygote which has got the combination of X and Y will develop into the male zygote, and the thing is by the female. So the sex is determined by the male Y chromosome, not by the X chromosome. So this is what given in a small paragraph in your textbook, after fertilization, sex determination in the zygote. Now, sex determination, this is done by a technique called amniosynthesis. You know very well amniosynthesis, amniotic fluid, that is the embryo when it is developing, it will form secondary membranes, amnion, and there will be a cavity developed amnion. In that there will be liquid amniotic fluid. Within the amniotic fluid only, the embryo will be suspended with the help of the placenta, a long rope. The long rope is the umbilical cord. Attach the placenta on the uterus part, attach the navel part of the developing embryo. So this umbilical cord, with the umbilical cord, the developing embryo will be suspended in a saccular structure, that is amniotic cavity filled with amniotic fluid. Now, roughly to say that, if you take this is the area where the placenta is there, and this is the umbilical cord. Now the embryo will be suspended like this, a head and the tail, and surrounding that there are amniotic fluid, a saccular structure, be fully filling up the uterus. This is the amniotic fluid. Now the covering is known as amniotic sac. Now they will syringe out this fluid. They take this analysis of the amniotic fluid, but they are able to identify the sex. But it is illegal now. It is banned, because when they are able to identify the sex of the developing embryo, but they are testing the amniotic fluid. You know very well, female infanticide is common still in India, in northern part of North India. So this is illegal. So it is banned. But this amniotic fluid test also helps you to identify sex-linked disorders. So there are two uses of this is called amniocentesis. Amniocentesis means testing the amniotic fluid. What is amniotic fluid? There is a saccular structure or a liquid surrounding the embryo. The developing embryo is suspended by a cord, this is umbilical cord. This is placenta. Now this is the embryo. Now this is amniotic fluid, amniotic sac. So when you are analyzing the amniotic fluid, they are able to identify the sex of the child, a developing embryo, as well as there is certain genetical disorders can be identified. So for identifying genetical disorders, it is positive. But the female infanticide is a common social evil at present in India also, in the North India. There are so many people, they don't prefer the female child. So that discrimination should be avoided. For that, it is made banned. So this technique is known as amniocentesis. Now that is sex determination, amniocentesis, both go together. What are the positive effect of why it is a negative result of amniocentesis is they are finding out the sex of the embryo, and it will lead to destruction of the embryo. They may abort it out. They found this is a female. When does the sex organ develops in the development of the embryo? Now the first day, second day, third day. That is third month, 12th week. First month, second month, third month. Third month of the pregnancy is known as the first trimester. That is 12 weeks. At that 12 weeks, all the organ systems of the body of the embryo will be fully developed. The limb buds and digits would have been formed completely. One miniature baby you will find. If you see the aborted embryo of a third month embryo, complete miniature. A head will be there, small thin limbs, and the digits will be there. Fingers and toes you can easily make out. And it will have the external genitalia will be fully developed. So now this sex determination, and again, if they find out, they should do it before the third month if they want to remove that. Because abortion or removal of the embryo, the first trimester or first third month, it is safe. After the fourth month, removing the embryo, that's called abortion, or removing the embryo, medical termination of pregnancy, MTP we call that as. Now it is made legal at present. Before it was illegal on 15-20 years back, but it's made legal to cut out the population growth. That is the next lesson you will be studying about the reproductive health in your 12th standard. That is MTPD, medical termination of pregnancy. That means medically you are allowed legally to terminate the pregnancy. What are the situations? One of the situations, this can be, suppose they found out the baby, they can approach the doctors and eliminate the embryo. But this should be done by 3rd month or before 3rd month. After that, it is dangerous for the mother body. It is not easy. Only thing is they won't surgically remove that. They'll make the uterus. Suppose the person wants to eliminate the embryo after the 3rd month or 4th month. Some people even 6 months, and when that is too stupidity, and they should have been more careful. And if in that month they want to eliminate the embryo, they have to induce the uterine contraction by injecting directly onto the uterine muscle. They will inject the hormones which stimulate the contraction of the uterine muscle, and they make the uterus contract medically, induce a contraction in the uterus, and normal delivery like they have to. So it will be more painful than the normal delivery. So now this medical termination of pregnancy should be done. This is one of the reason for amniotic amniocentesis. One of the reason banned is it may lead to the abortion of the female embryos. So this is about sex determination and amniocentesis. These points are connected to the fertilization. After fertilization, when does it talk about all that we saw, including the new plants? Now, next after fertilization, what happens? Now, the development, the embryo develops and the fertilization occurs in the ampullary region or the meeting of ampulla and isthmus. After that, the zygote does not remain inside. Automatically, it will be pushed forward by the peristaltic movement of the oviduct as well as the ciliary movement, and it passes through. The next part is what? Isthmus. So, after fertilization, what will be the next one? Development. So after fertilization, what will happen? Now fertilization. What is the result of fertilization? Zygote. Fertilization. That results in the zygote. Now zygote begins to develop. Now the first division of the zygote, development of the division of zygote is called cleavage. Cleavage in a sense, cutting. He didn't give you particularly the mitosis. You know why? It's a mitotic division. But what's the difference between cleavage and mitosis? When a particular cell undergoes mitosis, take onion root tip. And if you take mitosis, is easy to study in onion root tip. If you take an onion root tip to study the mitosis, you know very well they are meristematic tissues, and these cells are capable of repeated division. They undergo mitosis. When they undergo mitosis, what will happen? The mass of the cell increases. So particularly that organ increases in size. But in the case of cleavage, though cleavage, the mitotic in a sense, the number of chromosomes do not get reduced. The same replicating division. The zygote has got 46 chromosomes. Now the pieces also will have 46. The mitotic division. But it will not increase the size of the zygote. That is a single cell, a single zygote keeps on dividing, but this division is called cleavage. It's not called mitosis. Main difference is mitosis may increase the mass of the cell. That is the size of the cell, or the quantity of tissue may increase. But the cleavage does not increase the quantity of the tissue, neither does it make the zygote bigger. The zygote size will not increase. Instead of that, the number of pieces inside will increase. See, zygote is microscopic. When you draw on the board, you should not imagine such a big zygote is there and cut it into 2 pieces, 4 pieces like that way. So why the cleavage versus mitosis? If you take, you should keep this in your mind. Mitosis may lead to the increase in the mass of the tissue. Cleavage does not increase the zygote, but both are mitotic. Second thing, cleavage is very, very methodical. Up to certain stage, it has got a definite plane of division. Now, mitosis, it's not like that. You won't have a definite plane. It should divide longitudinally or horizontally like that. But cleavage has got a particular plane of division. For a plane of division will be methodical up till till the zygote cell divides into a group of 32 to 16 to 32 cells. Normally 32 to 64 we'll say. That is a single cell that will divide into 2 and then into 4. See, if you see the zygote division, it has a definite plane. Alright, and this cleavage, the mitosis will keep on growing. You take my plant tissue, meristematic tissue is alive. If the meristem is alive, only root will grow. And this meristematic tissue is capable of growing, dividing, and leaving the growth of the plant body continuously. But this cleavage does not increase the size of the zygote, and it has got a definite plane of division. The plane of division is there, and the number of division it is very regular plane. In the beginning, it is very regular, the division is. After that, the division will become haphazard in the sense, independent of one another. It will rapidly multiply. So the early stages, the division is very methodical. I've got a definite plane of division. It is regular division. Alright. Now next one is now the number of cells will increase only. Now definite division. What do I mean by that? Say suppose the zygote is there first. This is zygote. First it is divided into two vertically. Second division will be perpendicular to the first one. That is, first is vertical like this, second will be perpendicular to the first. So 4 pieces are formed. Now third will be horizontal, dividing that unequally, upper smaller and lower larger. Upper 4 smaller, lower 4 larger. So third division 8 cells. Alright, fourth division will be another one on top. That is 8 plus 4, 12 it will become. Then fifth division will be down. 12 plus 4, 16 it will become. Now after that, see, after this, you have the 4 directions in the north, south in your, the north, south, east and west. Now first is west to east. East to west, the right hand side, this is east and this is west. See, exactly like your the directions of the geography, east and west. Now first division is east to west. Second is north to south, perpendicular to one another. Now third one will be circumferential. That is, it will go round on the upper half and the lower half. Third, fourth. And the last one will be, fifth will be diagonal. And sixth will be diagonal. So, now the first division 2, second division 4, third division 12, 4 plus 4, 8, third division 12, fourth division 16, fifth division will be diagonal. 32, another diagonal 64. So that's what I meant. The plane of division is regular till it becomes a mass of cell like 32 to 64 or 16 to 32. So this division, finally, so the division, when does it occur? Now that is, as soon as after 1 day after the fertilization, just within 12 hours or 30 hours, in a sense, 1.5 day, 1 to 1.5 day after fertilization, the division begins. So it takes 1 to 1.5 day. In some books, it will be given 30 hours. 30 hours means roughly 24 plus, 24, 1 day, 12. And the other one, that is a real lesser than 1.5 day. In the book, it won't be given hour-wise, it will be given 1 day after the fertilization. After this 1 day after fertilization, the first division starts. So second division, this is the time, when does it start, okay? When does the cleavage start? And where does it start? When the zygote begins to move through the isthmus. Its ampulla, it comes. As soon as it comes the isthmus, it begins to start. So when this is the time, when does it start? When is the, say, 1 to 1.5 day, we can say 30 hours after fertilization. What happened? Now please close the door now. Sudha. What happened now? எப்படி பண்றேன்னு பாக்குறீங்களா இல்ல letters குட்டி குட்டியா தெரியுது நீங்க எடுங்க லைவ் you have to write bigger அங்க bigger எழுத சொல்றீங்களா it will become okay small okay they will be able to see okay and they want bigger. Let me repeat again, children, now fast, excuse me. So fertilization, okay. Now this is zygote. This leads to zygote. Now what happens to zygote? Zygote undergoes cleavage. Now cleavage, okay. Now when does the cleavage start? What is cleavage? It is similar to mitosis, but in a sense, the number of chromosomes are maintained. No reduction of chromosomes. That's what you mean by mitosis. But there is no increase in the size of the zygote. Mitosis will increases the number of cells and the tissue development will the organ will become bigger or develop. That's what I mean. But in this case, cleavage, the size of the zygote does not alter, and the cell inside the single cell, the nucleus divides surrounded by a little bit of cytoplasm like that. But each nuclear piece and the cells formed will have the same number of chromosomes. And the cells formed are, that is cleavage, okay. Now, cleavage is similar to mitosis, but it is not similar to mitosis. Similar to mitosis in the number of chromosomes, but it does not increase the size of the zygote. And secondly, the cleavage, the product we don't call them, they are called blastomeres. Blastomere. If it is a bigger one, it is called macromere. If it is a smaller one, it is called micromere. Thirdly, the cleavage has got a definite direction. Direction and plane. In other words, it has got a definite direction for the plane of division. First is vertical. I told you, if the plane, if you learn the directions of the geographical direction, first is vertical, first is straight, vertical, meridional. Second is perpendicular. The third will be horizontal. The third will be a little bit above the midplane. So upper smaller and lower larger. The smaller ones are micromeres and the larger ones are called the macromeres. And the fourth one will be around, again horizontal, circular in the figure on top. So finally it goes on till it reaches the stage of 16 to 32 cell stage. So when does it occur? When does it occur? When does this occur? 1 to 1.5 days. We can say the next day, some books it will be given 30 hours after fertilization. Now where does it occur? When the zygote begins to move through the isthmus. You know very well, the fertilization occurs just the previous part, that is ampulla. From there it comes the narrow part isthmus. When it travels to the isthmus, the division begins. So the first day, now the number of divisions if you take, now the first day, now you will have the blastula. The first day there will be 2 pieces, 2 blastomeres. 2 blastomere. Okay. Now the second day it will go to 4, and third and fourth day. Third, this is day, it's not a month. Now because we will see the development of the embryo in the month, so you should not muddle with the first, second, third. This is the first day. First day after fertilization, when the division begins, there will be in the 2 pieces. And the second day, it will undergo slowly. The second division is slow. It will become first 3 cell stage, then 4 cell stage. Now, so 4 cell stage. Now, third to fourth day, these 2 days, it will have a mass of about 5 to 12 to 16. By the time 7th day it comes, okay. Now 5th day about 16 cells will be formed. Then this mass of cell is known as mass of cells, which should I say mass of blastomeres. Mass of blastomeres form a ball-like structure. Now these ball-like structure, this is a zygote. Each one is there. Now this mass of cell, this ball-like structure is called morula. Each one is a cell. It has got a number of nuclei. In this, if you take, the central cell will be little larger ones, and peripheral cells will be the smaller ones. Outer side will be smaller ones. Now this ball of cell is called morula. Morula or blastula. Now morula means it's a mulberry. It resembles like a mulberry fruit. That's why it's called morula. Morula represents mulberry, resembling the mulberry fruit. So the word comes from morula, from the mulberry. Now it is nothing but a mass of cell having a central larger cell surrounded by the smaller. Why? When the division occurs, I told you third division, horizontal one, it is unequal. It's not exactly in the middle, equatorial plane, little bit above. So upper half will be smaller cells, lower half will be larger cells. So when they multiply, the upper half cells overflow, cover the outer side, and the lower one occupies in the middle part. So the entire blastula will have a mass of cell surrounded by a larger one inside and the smaller one out. Now surrounding this is zygote. Now what is the zygote? You go back to your primary tertiary follicle, cell, plasma membrane, then vitelline membrane, then zona pellucida. So zona pellucida is still there when the medulla forms. Zona pellucida, you have to reflect back your tertiary follicle. Tertiary follicle, as I told you, tertiary follicle, secondary oocyte with the polar body. Tertiary follicle, secondary, and it is started, meiotic division is arrested in metaphase 2, and the cell is surrounded by the plasma membrane. Outer to the plasma membrane, vitelline membrane. Outer to the vitelline membrane, there is a perivitelline space. Surrounding the perivitelline space, you have a uniform homogeneous zona pellucida, outer only corona radiata. So the zona pellucida remains when during the blastula stage, morula stage. So the zona pellucida surround, there will be perivitelline space. So this is the zona pellucida surrounding this. So this roll of cells will be 16 to 32 cell stage. It will be formed. Okay. So now all these are blastomeres. You don't call them as a cell. They are blastomeres. The larger one are called macroblastomere, micro one, macro. And the larger one occupies the center, smaller one occupies the periphery, but it is surrounded by the zona pellucida. Now this is a stage, the blastula, the morula rolls and rolls, comes to the now 5th to 6th day. On the 17th day, what will happen? This blastula will attach it, this blastula will go and attach it onto the uterine wall. Now uterine wall is already under the control of your, very well, Graafian follicle rupture. And if the fertilization has occurred, the mature Graafian follicle will develop into the corpus luteum. Corpus luteum will produces progesterone as well as estrogen, and they make the uterine wall much thicker, and the uterine wall is ready to receive this blastocyst which is coming down, blastula which is coming down. You follow? Now this blastula, when it comes to uterine, it will stick onto the wall of the uterus. Which wall of the uterus? Endometrium, innermost wall. So when it enters into the inner endometrium, these cells, the outer cells, now out of the zona pellucida, what will be the corona radiata, the outer covering only. Now corona radiata, all that will be dissolved by the time the sperm enters in. So this, when the blastocyst attaches under the endometrium, it begins to destroy the cells of the endometrium where it sticks on. So in the endometrium, practically, the endometrium is epithelial lining layer, glandular epithelium. So this epithelial layer begin to be autolyzed, destroyed, and it looks as though there is a cavity there in which this blastula is attached onto that, sticks onto that. Then this outermost layer, now up till that. So now this is about where does the fertilization occur, the further development. Okay. Now you go from here to there. So the this diagram is there already in your textbook. If they have given you a fallopian tube and the first cell stage all along the fallopian tube, first day, second day, third day, fourth day, and the blastula, the mass of cell is called the blastula, or it is also known as morula. It is also sometimes referred as blastula. It's also known as blastula or morula. Now this enters in the uterus. When it enters the uterus, the uterine wall is there. Imagine this is your fallopian tube. Okay. Let us take this is a uterine wall, endometrium. Is an endometrial wall, and so fundus of the uterus, body of the uterus. This is a fallopian tube. Okay. Now the morula has this morula has come and sticks on here. When it sticks on here, what will happen? The morula attaches onto the endometrial wall. When it comes in the endometrial wall, it will erode. The endometrium will be eroded. It will be like this. The whole place will be eroded out. It will be eaten up. It will appear as though it's like this. This is the place where the endometrial tissues is destroyed. So this blastula will be sticking onto the cavity like in the beginning. Then what happens in the blastula? Now the blastula has got now the inner larger cells, these cells, the inner ones are larger cells. Outer cells differentiate. They differentiate. The outer cells become spindle shaped and they are called the trophoblast cells. The outer cells little bit elongated. Now this is the trophoblast cells. Now the other zona pellucida here. Now these outer cells, the trophoblast cells, begin to poke into the form small finger-like projections into a villi. It grows into the villi. The outer membrane grows. This is the trophoblastic cell. Trophoblast. Then the inner larger macromere or larger cells. Now these are the smaller cells. The smaller cells differentiate into trophoblasts. These trophoblasts develop in the form of in the next stage what will happen? This is the endometrial wall. First it is forms a dent. Now this is a blastula. I'm not drawing the detail inside. The next stage what will happen? Now in the dent, this is a uterine wall. Uterus, this is uterine wall. That muscular tissues here. Now this I got a glandular cells and the follicular cells, blood vessels and everything. Yes, sir.
Continue.
Now the blastula forms. It forms a number of finger-like projections protruding into the thing. Now these finger-like projections formed by the outer layer, that is the trophoblast, is called the villi, chorionic villi. Chorionic villi. Now this is called chorionic villi. Why does the word chorionic villi suddenly are using? Chorionic villi. Now the inner cells will become an embryonic mass. Anything is wrong. Now the inner mass forms the embryonic mass. Now the surrounding smaller cells, which becomes a trophoblast, does not involve in the development of the embryo. It forms secondary membrane. These membranes are called amnion and chorion. Now this further development will be in the embryogenesis. You are not being explained there. So this chorionic membrane, the membrane developed from the trophoblast, is called the chorionic layer. This chorionic membrane forms a villi which will be poking into the uterine. It's small finger-like projections. It will be vegetating into this. So what is here in this place? Now, the uterine tissue is already eroded, and this is like this uterine tissue. There are no epithelial cells. Now what will happen? Something is wrong, sir. Now you have got the uterine tissue everywhere here. So this chorionic villi comes in direct contact with the uterine tissue. The uterine tissue, what will be the blood vessels and follicular cells, the secretory cells. So the chorionic villi interdigitating with this is the maternal part, which I am showing you. This is the maternal part. So this is the uterus. Say this is a uterine wall. The uterine wall it forms, erodes the uterine wall, the blastula, and this is a maternal tissue. Here is a maternal tissue, endometrium. This is myometrium. This is myometrium. Don't go there. Now the endometrium has got the blood vessels and follicular cells. So these chorionic, the developing embryo, the developing embryo forms. The developing embryo, that is a blastula. It has already become a inner mass of small larger cells and outer cell. Outer cell is called trophoblast. The trophoblast develop the membranes known as the chorionic membrane. The trophoblast gets involved in the development of secondary membranes called chorion and amnion. So this chorionic part forms, it develops into a small finger-like projections, now which will be eroding into the maternal tissue, pushing into the maternal tissue in the form of villi. So these villi are called chorionic villi. So what is the part in between? Now what is the portion in between? This is the maternal tissue. Now this is the maternal tissue all over. The cells are already damaged. So what will be there? All the maternal blood vessels will be in close contact with the chorionic villi. This one is an embryonic proper. This is a maternal proper. So this place, the chorionic villi, the blood vessels of the embryo, the developing embryos there. So the blood vessel of the embryo, the chorionic membrane, then the blood vessel maternal part of the uterine part comes. So this whole area where the embryonic part, membrane is a barrier, like how we get in the alveoli, the diffusion membrane, and in the case of nephron, filtration membrane. Now similarly, the barrier, this is called placental barrier. What is the placental barrier actually? Now the chorionic membrane forming the chorionic villi. Inside the chorionic villi, what will be that? Here is a maternal blood vessels and maternal tissue, mainly blood vessels. And here is the thin membrane, single layer of cell. Then you have the embryonic part. This is the embryonic part. Here you have the embryonic portion, the embryonic blood vessels here. So this whole thing, that is maternal blood vessel, the membrane, and the fetal. Fetal means developing embryo. The blood vessel is the barrier. Everywhere in your body, when you talk about the period, there will be 3 layers. You take alveoli. What are the barriers in the alveoli? Wall of the alveoli, single layer of alveoli. Then capillary wall, basement membrane. Then capillary blood. So nephron, you take the nephron, Bowman's capsule, the squamous epithelium or filtration pit, capillary basement membrane, then capillary epithelial cells. So there will be basement membrane, 2 cells on either side. Here you have got the chorionic villi, maternal tissue and paternal tissue. So what this area only is then the maternal tissue. Now what happens to this? There's an exchange between the maternal and the fetal content. What is this entire place where it is fixing on to one another, interdigitating with one another, that part is called placenta. So placenta is nothing but anchoring of the embryo through the chorionic villi into the maternal tissue. What is coming in close contact? Blood vessel of the embryo coming in close contact with the blood vessel of the mother or maternal blood vessel through a single layer of the barrier, that's the chorionic wall of the chorionic villi. So through that, the maternal blood, whatever waste material, particularly excretory product and CO2 from the blood of the embryo, is transfused or by diffusion goes into the maternal blood and it's excreted to the maternal body. At the same time, the maternal blood, nutrients, whatever is necessary, nutrients and hormones particularly, whatever the mother eats when she's carrying the baby, everything will be reaching. So when they have any fever or anything, they consume in medicine, that will also go into the embryo. But any item which they consume when they are carrying the child, it will be automatically, it will go to the developing embryo. So that is why the nutrition of the mother, the pregnant woman, is more important than having a child. If you are looking after the mother during the pregnancy properly, the female figure, the child will be a healthy child. So if you want to have a healthy child and have an easy life, you have to look after them. That is why now the government has come, the maternal, all the cares are nowadays. Pregnant women care later on, now recently only after last 20 years back, 15 years back. Special attention has been given to the pregnant women. Now, because the mother, this placenta, this tissue is known as the placenta. This is called chorionic placenta. Now, because of this, through what is the work of the placenta? Exchange from the mother to the embryo, and whatever the waste, what are the major waste material given in your textbook? Excretory product and CO2. So why am I telling you this? This is all for a NEET. Essay question, you can write a big story. Whatever I have given, you can put it in a paragraph and finish it there. Now, but actually, how the NEET questions can be there. So that is why you have to be very clear. The placenta is a combination of embryonic part and the maternal. What is the maternal embryonic part? Chorionic villi, blood vessel of the embryo, blood vessel of the mother, or maternal blood vessel, separated. Now, this placenta is where, see, the whole thing is uterus. Only this place, wherever the anchoring of the blastula occurs. Now, before the anchoring of the blastula, what will happen? The blastula will, this stage will become a blastocyst. I didn't tell you the blastocyst. After that, only the implantation will be there. Now, before the implantation, 1 second, please. Now, this is wow. See, before it is forming, this is a modular stage, okay? Now you have this is a modular stage, full of cells. Now this modular form, you have the inner larger and outer smaller, trophoblast and the inner one. Now this trophoblast and the inner one, I start from there. You have got the trophoblast outside. These are trophoblast, smaller cells out. Trophoblast are smaller cells. And in the inner cells are the larger cells are inside. Okay. Now larger cells are inside. The trophoblast absorbs the uterine liquid. It is in the uterus, am I right? This implantation in the uterus. The wall of the uterus produces the nutritive liquid, and the trophoblast automatically, this morula absorbs the nutritive fluid of the uterus, and the fluid enters in. When the fluid enters in, now there is a cavity develops in the blastula. The blastula, which is a mass of cell, develops a big cavity inside. It forms a big cavity, and the cavity, when the cavity develops, now these central cells are pushed to one end. Now the central cells move to one side. Okay. Now the peripheral cells, these cells form the rounding about like this. So during the blastula, is a mass of cell. There, the inner cells are larger, outer cells are smaller. I have to show the difference between these two. The inner cells are larger inner cells. These are larger inner cells surrounded by smaller one. So when the next stage what will happen, the larger cells go to one corner, one end, they are pushed to one end, and the other one, a cavity develops, and the smaller ones form a surrounding around that. The smaller cells form a thin covering around this. So the outer one is known as the trophoblast, inner one is known as the embryonic mass. Now these are the cells, okay? So this one is the outer one is known as the trophoblast, and inner one is known as embryonic mass. Embryonic mass. Embryonic mass. Now this cavity is known as the blasto seal. Blasto seal cavity. So when it forms an implantation, it has got the blast. Then this trophoblast only forms the villi. You understand? So this condition is known as, this is blastula, this is blastocyst. This stage is known as the blastula, this is blastula. I'm sorry. Blastula, mass of cell, this is blastocyst. Blastocyst anchors. Forms a, so the trophoblast outside will form secondary layers, I told you, chorion, secondary membrane. This chorion forms a villi and then implantation occurs. This fixing up of the blastocyst onto the uterine wall is known as implantation. You have to know the term. Implantation means options will be given. Now up till this, now when let me finish this, then we'll go to the objectives, how much it can be. So the blastocyst anchors onto that. This place is known as a placenta. Where is this area? You have an embryonic part, maternal part, and the chorion membrane, chorionic villi. This whole tissue is known as a placenta. What is the function of placenta? Exchange of the nutrient and the things from the mother, and the waste products from the embryo is expelled through the mother's blood. So placenta is the functional and structural unit of a developing embryo. So the word functional and structural unit is given. You will come in zoology many places. Functional and structural unit of brain tissue is neuron, single cell. Structural and functional unit of the muscle, sarcomere, distance between the two Z line. Functional and structural unit of a kidney, nephron, it's a structure. So this functional unit will be there in many places in your zoology. So here in this lesson, the functional and structural unit of a developing embryo is a placenta. The word is there in your textbook. So you have to be very careful for your objective. That's why I'm insisting on that. After the implantation occurs, what happens? Then endometrium. This endometrium, it goes around the blastocyst. Completely, it covers the blastocyst, the inner wall. So the whole, this is the implantation, and the whole blastocyst covered by the endometrium fixed onto the thing. Now further development is the enlargement. Now in the beginning, now this is wall, this is space inside. Okay, the chorionic villi will be everywhere, all over the body. Now I am only showing this. In the early stages, the chorionic villi will be formed around the entire blastocyst. But later, the one which is facing interior of the uterine will disappear, only which is anchoring will be here. So this placenta, so this embryo begins to grow bigger. Now when it becomes bigger, it will occupy the entire space here. So when it becomes bigger, the tissue, with this placental tissue, the outer chorionic membrane and amniotic membrane, it forms a long rope-like structure that is called umbilical cord. So when this one develops, it's a uterine wall. So to repeat back again, before I clean the board, first day 2 pieces, second day 4 pieces, third to fourth day 8 to 12, then 12 to 16 by 15th day, 16 to 32 cell stage, the 6th to 7th day. The 7th day when it comes, it becomes a blastocyst. That is, when it becomes a mass of cell, it is called blastula. That is on the 15th day, it will be like a 5th day, it will be a 3rd to 4th to 5th day. You cannot simply say delimit, 4th day it should be like that, 5th day it should be like that. No, it's a continuous process. So now by the 4th to 5th day, it will be a mass of cell. That mass of cell is called blastula. And this mass will have the largest cell in the center and the smaller cell in the outside, because the division after the third division is unequal. The upper smaller and lower section will be larger in the case of zygote. So the larger cells and smaller cells. So the central cells will be larger cells and the surrounding cells will be smaller. Smaller cells occupy the surrounding, but the zygote is still surrounded by the zona pellucida. That's the original covering of the ovum. Now then, this the outer cells. Now the outer cells go on the outer lining layer. The outer layer, what happened? The trophoblast, this is in the uterus. It absorbs the nutritive fluid in the uterus. Now this fluid begin to accumulate in the center, pushing the cells, the central cells, to one end. So the central cells, the larger cells which are pushing into the one cell, become the embryonic mass, and there is a cavity filled with the fluid, and the trophoblast surrounds the cavity. This condition where the ball of cell develops a ball with an empty space inside, that is called Blasto Seal, and that condition is called blastocyst. Sometimes the Blasto Seal itself is known as blastula, but Blasto Seal is a space in the morula. That is no longer is a mass of cell, there is a gap. Like in a tertiary follicle develops an antrum, similarly here. So the Blasto Seal, how this Blasto Seal forms? The trophoblast outer layer of cells absorb nutritive fluid which is secreted to the uterine. These fluid begins to accumulate inside, pushing the larger cells to one end, and the central the liquid filled space is known as the blastocyst or blasto seal. Seal means cavity. Okay, now the cavity of the blastocyst is filled with the liquid, and automatically the trophoblast will be pushed outside. That forms a lining layer. All the larger cells forms the embryonic mass towards one side. So the embryonic mass is on the surface. You have got the trophoblast, and the blastocyst also surrounded by the trophoblast. This outer trophoblast produces secondary membranes. Now, what are the secondary membranes? Chorion and amnion. Now, these secondary membranes only fold in the form of they develop in the form of finger-like projection. First, it will completely erode the inner endometrial wall. It will be like a cavity onto which this goes on. Then this eats up more of the endometrium and buries itself into the uterine tissue. So the uterine tissue, when it buries itself, the finger-like projections are surrounded by maternal blood vessel. So through the thing, by the time the embryo, the embryonic mass gets differentiated, it begins to develop into the part of the embryo. The further development of the embryonic mass, you are not ectoderm, mesoderm, germ layers will be formed. 7th day, implantation. So one of the questions will be now. I'll finish up this, then we'll come to the question. Now, when it is implanted into it, the whole area is known as the placenta. Now, this fixing is called implantation, and the entire blastocyst will be covered with a endometrial lining. Okay, now this placenta is the functional and structural unit of developing embryo. Now, what are the questions in this can be in your NEET point of view? First, we saw up to fertilization, everything. After fertilization, when does the cleavage begin? One other question. Options I can give anything. Ampulla, infundibulum, fimbriae, just generally oviduct. I can give that. In that case, actually the cleavage begins when the zygote moves towards passes through the isthmus. When does it begin? Immediately after 1 to 1.5 days after fertilization. 2 cell, 4 cell also slowly. 2 cell will become third and then only 4 cells stay. Then by 3rd or 4th day, it will be 12 cells. By about the 4th to 5th day, it will become a mass of cell that will be 16 to 32 cells. Now, this is called morula. By the time it comes to morula, it has come the end of the oviduct, which is in the uterine wall. So, they can ask you, now what are the changes? When does the cleavage begin? And what is the difference between the cleavage? You have to be very careful. It's mitotic, but the planar division is regular. The cleavage that continues only for till it anchors only the cleavages. Till the blastula formation only cleavage. Once it becomes a blastocyst, no more cleavage. It becomes the figure, everything, the changes, alignment of the cells change completely, arrangement of the cells. So when does the blastula develop? About 4th to 5th day. Now the days are there clearly in your textbook. If it's not, the diagram is there in your textbook. But the days are mentioned, not particularly 2nd day, 3rd day. But he has given by the 7th day, the blastula has developed already into a blastocyst, then implantation occurs. Now, how does the blastocyst develop? The first question is the day, where does it cleavage occurs? Now, what is the cleavage? The division of the zygote is known as the mitotic division. or amitosis, or meiotic division, or cleavage. So cleavage is the word only used for, that is one option. Statement questions can be there. Statement questions, how can I say cleavage is identical? It is identical to mitosis. In which are the ways it is similar to mitosis? An option they can give you statement, which is a wrong option, or which one is the correct option. And a cleavage can be similar to mitosis in the nuclear distribution. But mitosis will lead to the increase in the size of the tissue that doesn't occur. So cleavage occurs only for a short span. It's not lifelong division of the growing tissue. That one statement. If you know the facts related to the cleavage, you will be able to find out which is a wrong statement, which is a right statement. And cleavage is in a methodical plane. The division occurs in a methodical plane. Now that is, mitosis is not like that way. Then you will find mitosis, prophase, metaphase, anaphase, the stages, and the karyokinesis, cytokinesis. That doesn't, it's like a fission. But it cannot be taken as a fission because DNA pieces, every piece will have the same number. Thirdly, cleavage does not increase the size of the zygote. Remember that. So if you know those facts, statement question can be there. Now, objective question also we can ask. How the, but in this cleavage and mitosis statement is more expected than the multiple choice. Then the blastula, mass of cell. A mass of cell from the zygote produced. They can ask you blastula, blastocyst, zygote, none, or some other option. So the mass of cell without any cavity is the blastula. Another question: when does the implantation of the zygote occurs? The zygote occurs implantation at the stage of morula, morular blastula, or blastocyst, or embryonic stage, or fetal stage. So then occurs only in the blastocyst stage. Now the blastocyst is the outer covering. Then they can ask another question. And the mass of morula, the cells of the morula differentiate into embryonic mass and trophoblast, or amnion or chorion. Some other names they can give you. So that embryonic mass is different, trophoblast is different. The cells of the blastula differentiate into embryonic mass, central embryonic mass, periphery by thinner cells are trophoblast. Then blastocyst and blastula difference. The difference between the blastocyst and blastula. Blastocyst has got a cavity and the blasto seal, and the embryonic mass pushed separately, surrounded by the trophoblast. This is one question. Then what is placenta? Functional and structural unit of the developing embryo. Famous, multiple choice they can give you. And what is implantation? Fixing up of the, they can ask you fixing up of the zygote, or fixing up of the blastula or blastocyst. So only the blastocyst fixes onto that. And the blastocyst is covered over by the, completely covered by the endometrial wall. This covering of the endometrium is given, Paka in your textbook it is given. So this is about the implantation and placenta choices. Now after this what will happen? The embryo begins to develop, and the whole when it develops, it occupies the entire space in the uterus. In that case, now you have. So I think you can go over this part. I can tell it. Then you have. If you take the uterine cavity, say this is a uterine wall. Take it as the uterine wall. Now in this area, the placenta area here, placental area, I'll show you that. Then umbilical cord, and there are secondary membranes and amniotic cavity, and the hanging embryo, the head, and the whole will be like this, a tail end and a head end. Nicely you can make out. Embryonic development. Now this is a uterine wall, where you have got, so this is the wall of the uterus, okay? The tissue of the uterus. Now this is a tissue of the chorionic, the placental chorionic villi inside. Chorionic villi. And the maternal tissue in between is the placental area. Okay? This whole place is the placenta. Now this is the umbilical cord and embryo. This is placenta, umbilical cord, umbilical cord, umbilical cord, umbilical cord, umbilical cord. Cord is a rope. Cord means rope. Now this is the embryo. Embryo. Now this is the amniotic fluid. The fluid, amniotic fluid. Amniotic cavity, amniotic fluid. So the embryo. Now take out the uterus. This is the whole of the uterus comes, and lower part will be the cervical canal. So the embryo, as it develops, see, this is a liquid. So the umbilical cord, the embryo is constantly, motion of the embryo is important. The embryo begins to move, movement of the embryo, and again, the next part of it is development of the embryo. Now what happens what happened the first month of the development, second month, third month. Now third month is first trimester. Trimester means third month. Second trimester means 3 6 months. Sometimes suddenly they'll say first month and second month, suddenly they'll say 12th week and the 24th week and 30th week. So when they give the week, you just mentally take it as easy to understand, easy to calculate the first month what happened, second month what happened, third month and third, fourth, fifth and sixth. So first month is actually heart is the first organ which develops when the embryo is in the first month. This is another question for a NEET oriented question. This I hope I can remove this modular stage. Now first month what happens? The first month of the development, heart. So now after, that will say after fertilization within 1 or 2 months, zygote business, 7th day fixation, already fixed, placenta form. So 7th day means first week after conception. Now first week, second week, third week, before they settle into the menstrual cycle, the end of the third week. Yeah, go to the menstrual cycle. Ovulation 14th day. Now within 16th day conception. Then you take, it goes on. 7th day implantation already. You are coming to the end of the month. So your menstrual cycle will begin. So automatically, how will the individual will know they are pregnant or they have conceived? The menstrual cycle will skip. So automatically, they will have a doubt that is whether they have conceived or not. So the test will be done. As soon as they skip the menstrual cycle, the test will be done. You can do the test. Now, these pregnant, when they're pregnant women, now before I go to the development, one more thing which we have to do. I'm sorry. Now this, I'll go to this month. Before that, some of the placenta, function of the placenta. The placenta produces a hormone. It is not only a structural and functional unit. It's a main. What is the use of placenta? Fixing the embryo to the uterus. That's one. Lodging. Second is it acts as an endocrine gland, endocrine organ. The most important endocrine hormones which are already here in your book, textbook, it is given. Human chorionic gonadotropin. Human chorionic gonadotropin. Human chorionic gonadotropins. And human chorionic lactogen, right? Progesterone, estrogen. These hormones are secreted by this placental hormones. Now the placenta acts like a endocrine gland, endocrine organ. Now what is the use of this human chorionic gonadotropin? This human chorionic gonadotropin acts like a LH. What is luteinizing hormone? Luteinizing, sir, it helps in the development of the corpus luteum. So this HCG, in the early stage, it is produced by the placenta. Later stage of pregnancy, it will release by the uterus itself. Yeah, it will develop in the ovary itself, it will liberate that. But in the early stage, the placenta produces the human chorionic gonadotropin. What's the use of this human chorionic gonadotropin? It acts like a pituitary LH and maintains the and allows the Graafian follicle called corpus luteum to produce progesterone. So this corpus luteum produces progesterone. What is a progesterone? It maintains the proliferation of the uterus and prevents it maintains the pregnancy. So the early stage, HCG acts like a pituitary LH, and that's one. Second thing, it helps in the maintenance of the corpus luteum and developing the progesterone, which maintains the strength of the uterine mucosa, endometrium, so that the implantation will be, the pregnancy will continue. Now, thirdly, now this hormone stimulates the interstitial cells. That is, the sex organs are developed, The sex organs develop during the embryo, the third week completely. All the organ system. Third month, that means 12th week. 3 was a 12 week. Third month completely is a sex organ. All the organ systems are fully developed. And now you know very well in the male specimens, there's a descent of testis to the scrotum. Now the HCG stimulates the interstitial cells of the testis to produce androgen and helps in the descent of testis to the scrotum. So the human chorionic gonadotropin helps in the maintenance of pregnancy, acting like a pituitary luteinizing hormone. So the LH maintains the corpus luteum. Corpus luteum maintains the production of progesterone. Progesterone maintains the wall of the uterus. So that pregnancy will continue. So that is the main function of this. And estrogen and progesterone. Now, during latter part of the pregnancy, another hormone called relaxin. The latter part of pregnancy, relaxin is produced by the ovary. Now these three hormones, HCG, HCl and relaxin. Now these three are called, produced only during pregnancy time in the individual, so they are called pregnancy hormone. Alright, this is one of the questions. Invariably will be that. Now they may give you any one of this. Any question I can ask you. Relaxin is produced, puberty, adolescent, pregnancy, parturition. They can ask smartly up in any manner. You have to be very clear. This is pregnancy hormone. That means during pregnancy only these are produced. Now then another one, HCG, which is a pregnancy hormone, acts like anyone. Helps in the ovulation or maintenance of the blastocyst, anything they can muddle up, multiple choice. You should know it acts like a pituitary LH. What is a pituitary LH? Maintaining the corpus luteum. So to produce progesterone, that is necessary for maintenance of the pregnancy. So these three hormones are called pregnancy hormone. If you are on a public exam, they can ask you this as a one-word answer. Which are the pregnancy hormones? You have to write the three hormones. Apart from this, during the pregnancy, there are other hormones in the body of the organ and mother body will show a lot of variation. That is estrogen and progesterone. Cortisols. What are cortisols? Cortisols are hormones produced by the adrenal cortex. And thyroxine, thyroid hormones produced by the thyroid gland. Now these hormones will occur in a larger amount in the body. That is another question. The hormones will increase during, why these hormones are increasing? Now what is cortisol? Mineralocorticals, glucocorticals and sex steroid corticals. Now the glucocorticals and mineralocorticals control the metabolism of the minerals and glucose. So that is necessary for during the development of the embryo and for the energy for the mother. So these are the general thyroid, basal metabolic rate. So that is necessary to maintain the metabolism of the maternal body. And if the metabolism of the maternal body is normal only, it can feed the embryo which is growing there. So the maintenance of the normal health of a female will depend. All these hormones will increase in there during the pregnancy. Two different questions. Which are pregnancy hormone is different. Another one is which are the generally hormones which will shoot in their level during pregnancy. So the mother body completely undergoes a metabolic change. The entire body system is not a normal after she conceives the child. During pregnancy is different from the normal days. Okay. Now during the delivery time, parturition time, oxytocin will be liberated. Oxytocin and will induces the contraction of the uterine muscle. Okay. Now the contraction of uterine muscle, back again it will allow the uterus to contract more. So during the delivery time, another hormone is secreted in the body which will increases the contraction. That's a reflex action like. The hormone secreted, in return the contraction increases. And the contraction increases, the hormone secretion will increase. It goes on. That's a reflex action like one into the other. That is necessary for the expelling the fetus outside the through the birth canal. Now this is the apart from the structure and part of what is placenta, how does it form? What is placental barrier? And what is the structural and functional unit of the developing embryo? Now how the embryo develops attached to the placenta, umbilical cord, placenta. What is the function of placenta? Now these are the areas you will have the multiple choice or statement questions. And the umbilical cord is the one which umbilical cord has got nothing but a little connective tissue full of blood vessels. A major between two ladies, like it, there will be two veins and one artery. as a characteristic of umbilical cord. We call that as a major between two ladies' leg. The umbilical cord, another thing is, the umbilical cord cells, this placental cells, umbilical cord, the tissues, there are stem cells. Now, recent study is preserving the umbilical cord, where they have the stem cells which have got totipotency. What is totipotency? It can develop into any tissue. You know very well, these tissues, stem tissues, are present at study in a research system for tissue transplant. For the diseases like your leukemia, blood cancer, bone marrow transplant occurs. So this is used. The stem cells are used for a tissue transplant for certain diseases wherever it is needed, because these stem cells have got the totipotency to develop into any tissue wherever they are implanted, where they were transferred. So the placenta umbilical cord is one of the source of the stem cells. You follow? It is now first during the delivery, they'll cut the umbilical cord, remove the child out, the fetus out, once it comes, and after that the placenta won't report immediately, because the contraction of the uterus should be more. So when the contraction of the uterus is more, then only the entire placenta will be expelled out of the uterus. The placenta itself will weigh 1 kg. It will weigh 1 kg. And the placenta has got a nice spherical structure. And the center will be a rope of blood vessel, and it will have a round, full of blood vessel network. Very glassy. If you have seen that in person, it will be very pretty if you can appreciate it. After that, it will be full of blood and everything. But once you clean it up and everything, the placenta is transparent, and you have to see whether the entire placenta is expelled, because completely it is full of blood tissue. So if the bits and pieces of placenta remains inside the uterus, it is not expelled out completely. It will become septic. It will lead to a high temperature. Immediately the patient will expire within 3-4 days. So during the delivery, not only removing the fetus, and the surgeons, the people who are conducting the delivery will wait for the placenta coming out entirely. Suppose if they have a doubt it hasn't come out, they have the curectomy. Curectomy means they will insert the instrument inside the uterus and scrape out the entire endometrium to make sure all the blood tissues have come out. Blood tissues will become septic if it is held up inside the body. So that is the main part in the placental. Placental hormone, which are placental hormone, which are the hormones increases during the development, which is the hormones is produced at the end of the latter part of the pregnancy and during the delivery. So these hormones, why these hormones are necessary? They change the metabolism of the mother, which is necessary to feed the developing embryo. Okay, this is up till implantation, cleavage. I hope we have made it very clear. Cleavage, blastula, blastocyst, implantation, placenta, placental barrier, function of the placenta, placental hormones in the body of the maternal blood. All right. Then next we go to the parturition. Parturition and mammary gland, I think. Now, the parturition, during parturition, what happens? Mainly, when the embryo is fully developed, fetus, now the completely developed fetus induces the parturition. That's a reflex action. Fetal reflex, that is called. Once the fetus is fully developed. So when it is rotating like this way in the liquid, see, if I have drawn, suppose you take like this, purely diagrammatic for you to understand. Now suppose, now that is the uterus, you have come to the uterus end like this, you have come to this end. Now what will happen here? Uterus. Now what happens to this? It becomes a cervical canal, neck of the uterus. Neck of the uterus, you know very well, you have the cervical canal, internal os, external os, then vagina. Now this is cervical canal. Purely diagrammatic. Don't imagine it be so crude like this way. Now this is the endometrial wall. That is a myometrium, endometrial wall, and the embryo is there, fully occupying the entire uterus as it grows bigger. When it is rotating like that, this is a problem when it is in a liquid. Now, before we go to this, what are the changes in the embryo? Now, first month of the embryo, as I told you, heart. This is there in the last paragraph in your textbook. First month, first organ to develop is the heart. So, after that, if the person is skipping the menstrual cycle, automatically they'll go for the checkup. All right, 7 days, first month. Now, this can be easily tested. The pregnancy test is, say, if they skip the menstrual cycle, they will be waiting 7 days, 8 days fast. By the time they come to the 14th day, they'll go in the middle of the month. That's the first month. So at that time itself, when you can, this HCG and HCl could be found in the urine of the pregnant mother. So they will confirm the pregnancy with the urine test itself in the lab. So by that they can easily tell you are pregnant or not. It will be confirmed by the end of the first month and the second month that they already skipped one menstrual cycle. If you go on the 14th or 15th day, automatically you will know how many days you passed. Even after the 1 week after that, you can make out easily. They can test and tell you the pregnancy. So the first month, see, you have to have a roll of this 14th ovulation in a menstrual cycle. If you take the month wise, I already told you first of the month to fourth of the month. First of the month, fourth of the month bleeding. Fourth to eighth is a post-menstrual period. 8th to 20th is a follicular stage. 20th to 30th is a secretory stage. So if the 20th is 30th on the secretory release is not there. So 30th of the month, the individual does not get the menstrual cycle on the first of the next month. So she is waiting for 1 week past. And then 8th to 20th, the follicular stage started. So she waits for up till 20th. It may come 1 week, 10 days, 10 days, 12 days. Now 20th or the 8th to 20th days or 15th or 20th of the month has come. When they go for the test, already 1 month is passed. Now 7th day itself is a fertilization is already this 14th. Go around, another 14th it has come. So when the 1 month fetus, already the heartbeat will be there. Now the first month of the fetal development, can I clean this part? I will remove. Now what? This is again textbook. I am only orienting on a textbook and a neat part of it. Now first month, what is the first organ to develop? Heart. So the first month, if the doctor says, they'll feel the heartbeat on the surface, the uterine surface. So they with a step they can make out the heartbeat. So it's very interesting to know. If you want to see the heartbeat of the daily embryo, you can easily see in a chick embryo. You take an egg. Now the egg which normally, when the fertilized egg only which we are eating as egg, chicken egg is a zygote what we are eating. You can incubate and the chicken will come. So that's a zygote. If you take the chicken egg, now suppose it is spoiled, sometimes they say spoiled egg. It remains for a longer time, the embryo will begin to develop in that chicken embryo. You can easily take the egg, there will be a broad end of the egg will be there, the egg will be tapering, it's for your interest only, I'm telling you it is like this, will be take. Then, now here, you should slowly, without damaging mass, you must remove the calcareous shell. It is remove the calcareous cell slowly, right on top. Here you will find a thick area, the embryonic disc, and a small dot and a blood vessel you can see. And if you look at the dot correctly, you can see the pulsation of the blood vessel. So the pulsation or the heartbeat is the first organ to develop in that embryo. Here human embryo, first organ to develop in the first month is a heart. Now the second month what happened? This is month, not day. Menstrual cycle is day. Now this is month, first month. Okay. Then the second what will happen? The limb buds will develop. Limb buds and the digits. Digits are fingers, finger tips like and the whole head part will be prominent. A jutting out area will be there. Now the second month, third month in your book it may given as a third month or 12th week. Now this is how they mix up. Now when you read the question, see in the question week is given or month is given. They may give you 8th week. Okay, now invariably the heart they won't ask you first month because everybody will know the first. Which organ to develop first is mainly heart. Now second one, the next is the limb bud. Limb bud will develop when the embryonic head this is the embryonic, if you take it, the head will be low will be like this way. Now the whole embryo will be like this only, completely, not like this. The head lobe will be there and the umbilical cord. Now this will be like this position. Now you will find the limb buds like this way. Now these are umbilical cord attached onto the navel region. The limb buds, development of the limb buds and the digits. Now the head will be ugly specimens when you see the embryo. Now you will find one bulge over there that's going to be developing into the eye like. So the limb buds and digits are second. Now by the end of third month or 12 weeks, all the organ systems of the body is fully formed. It's a miniature of human. If you have seen the third month about a child, you will find the head nicely developed. The eye will not be open. Nice round eyeballs will be there. Limbs will have developed. And the finger digits, all the digits of the toe as well as the finger toes, thin bones will be there. You can make out easily, and completely and the most important in this, all the systems are developed. All the systems or the organ is developed. Very prominently, you will find the limb, complete development of the limbs. And you can see thin bones like that. The digits as well as upper limb digits and lower limb digits will be fully formed. And the next one completely is the external genitalia. External genitalia will be formed fully. You know very well, external genitalia, scrotum, penis. The case of female labia majora, minora, clitoris. So the sex could be easily found out there. Now the 4th and 5th month. Now 4th, 5th, and the next development will be 24 weeks and 42 weeks. 24 weeks, 6th month. So now this is 3rd month. 4th and 5th month, further development goes on. Increase in the size. Okay. Now by the end of 6th month, the 5th month, what will happen? The scalp, head hair on the head will develop. Development of the hair on the head is prominent. Now in the 6th month, body hair, completely body hair. Now it won't be given as 6th month, 24 weeks. Very clearly it will be given. Now 6th month, see, body hair is different, scalp hair is different. The hair on the head develops before your body hair. You know the difference, the body hair is very thin and delicate ones. So the body hair, this is one of the objective questions. The 24th week of the development of an embryo, the organ which is formed. In this, body hair, eyelids will open, eyeballs will be fully developed, eyelashes will be open, eyelids will open, eyelashes will be developed there. So the eye part will open up and eyelashes. So head part occur completely. In this case, body hair 6 months, and it is given in your book. Eyeballs open and eyelashes. See, the latter part, first part of the reproductive system, easy. Attested you will study. Here only they can loop you because the points are very little there. The NEET questions, when you say multiple choice, invariably this question you should know, definitely. It is in the last paragraph in your textbook. You can write by the side of the paragraph, you write it down 1st month, 2nd month, 3rd month, 4th month, 1st trimester. Or sometimes they'll ask you, this is considered as the 2nd trimester. This is the one. This is 2nd trimester, or 6th month, or 24 weeks. Any manner they can. And don't think the body hair, the scalp hair develops more than before the body hair. Now, this is another mistake the children made when they do the test. So 24th week is a 24th week hair only is given, hair development is specifically body hair is given. So the external genitalia fully developed the 3rd month is the one where the full miniature human being is there. So this is another area where we can trap you for a multiple choice, a statement questions, or even master following I can get. 1st month, 2nd month on 4, 1st, 2nd, 3rd, and 6th I can take and give these changes. And cross master following can be asked. Do you follow? So this is the different months development. Now when you come to the parturition. Now when the parturition occurs, now mainly when does the parturition or delivery? Now delivery mainly when the fully developed fetus only starts this contraction. It's a reflex action. So that is the fetal reflex. Now, fetal reflexes, once the embryo is fully developed, okay, it automatically as a reflex action, it induces the contraction or the uterus. So, when the fetus is fully developed, imagine the amniotic is fully covered, the full area is covered. That is this amniotic thing, the developing embryo will be fully occupying the uterus part. After this, you have big one. The embryo is fully formed. Developed embryo. You have so cold this is. See the umbilical cord and the embryo is enlarged in its size, everything, all the development and everything is over. Umbilical cord and the amniotic cavity fills up the entire uterus like this. So when it is rotating in the liquid, the head portion will come and fix it onto the septic canal. So that means when the head is fixing onto that fixation of the head, you call that as. Now the doctors will, once the reflex started, only when the fetus is fully developed, it's called fetal reflex, uterus might begin to contract. slight contraction will start. The patient is complain of that. They will say the stomach pain starts. They say simply stomach pain. It's actually uterine pain starts. So the limit they will wait for the uterine pain starts means this will be still rotating and the head part should fix into the fixation of the head. They'll wait. So once the head part, this is cervical canal, internal os, external os, vagina. So the entire thing. Another question, which is the birth canal. Birth canal includes cervical canal and vagina, not only vagina alone. You know there is a fold in the vagina, so the whole thing stretches means the vagina can stretch out, expand out. So the canal can extend because of the fornix. The vagina is not a straight tube attached over here. It's not like that way attached over here. You follow? It has been folded into it. So because of the fornix of vagina, the vaginal canal can expand during the delivery time. Now what hormones? The parturition is started by HCl, HCl, some hormones they'll give you. It's a fetal reflex. Fully developed fetus is responsible for the beginning of the delivery. Okay, now that is called fetal reflex. Fetal reflex, one word will be given. They'll give you 2-3 hormones for a multiple choice. Now that induces a parturition or delivery. So you should take the fetal reflex. Now secondly, birth canal. Birth canal, they can give you dumb, uterus, vagina, cervical canal, plain vagina alone, cervical canal and vagina, or fallopian tube. Fallopian tube they won't give you, they may give you some absurd answers will be there. You can eliminate that. The birth canal includes cervical canal as well as vagina. Okay. Now, when the fetal reflex starts, the uterine contraction, the hormone oxytocin is released. This oxytocin contracts the uterine muscle. When the uterine muscle contracts, more oxidation. That oxytocin and the uterine muscles are in opposite direction. That is, they are reacting on each other. As more and more hormone produced, more and more the contraction will become violent. So when the contraction becomes violent, the whole thing is expelled out. That is, the rupture of the amniotic, the sac ruptures, and the whole umbilical cord is quite long, and the baby, the head will be fixing up. So the people will remove the embryo, cutting out the umbilical cord. Because it has to be cut and removed. So the remaining umbilical cord, the placenta will remain inside the uterus. Once the fetus is removed, they will wait for the contraction to continue, and the contraction will expel the placenta of the umbilical cord. Then the rest I told you what it is. Now when this is going on. Now the newly born baby should be fed by the milk secretion of the mammary gland, lactation. So the secretion of the mammary gland at that time, the newborn baby should be fed by the mother's milk, because the mother's milk, the first few days, the secretion of the mother's milk is known as the colostrum. You know that. Now that contains the antibody IgM. This comes in your chapter IgG, not M, IgG. Now, this immunoglobulin G, this comes in your immunity chapter, health and diseases, the same second volume. Latter part you could have studied immunity, health diseases and human diseases, health and diseases. There one immunity section will come. In that immunity section, different immunoglobulin, structure of the immunoglobulin, thick, broad band, thin band, and thick band, antigen antibody binding, different immunoglobulins. A, D, E, and G. Now this IgG is the IgG is liberated it is present in the case of colostrum. So this antibody induces natural immunity. Why am I telling you? Now this is another important question in your multiple choice. That is the colostrum. The child should be fed with the colostrum at the first few days. Milk secreted in the women after the delivery. It's a natural food. That is the most, and the children fed on this has got more resistance than the children that are weaned away. Weaned away means cut away from the mother's feed. Now, so why is it necessary? This first milk, the colostrum, you could have the same thing in all the mammals. You could have seen in the cow also. Now they use the cow's milk after it gives birth to the calf. They use that milk and that to use even the milk sweets they use to make. But it's not only that sweet part of it, it is very rich in antibody IgG, which gives you a natural immunity. So they have passive immunity. So this is actually the natural immunity is obtained. So they can give you this one a question. What is colostrum? They can. And why should the children be fed? Now what is the immunoglobulin there? Why it gives you a resistance? The reason is the presence of IgM. When you come to the other chapter, what is the use of IgM? They'll ask you. Now, what is the use of IgM, IgE, which otherwise they'll give you another question. Which immunoglobulin induces the natural immunity or it induces the reaction? So that is that when you learn the immunity chapter, you should know all the IgD, IgE, IgM and IgG. So what are the function of each immunoglobulin? This is one of the immunoglobulin present in the mother. Now, where is this milk produced? That period is known as the lactation period. The milk is secreted in the mammary gland. We didn't see the mammary gland. You know very well, mammary gland is a modified gland, skin gland. Now, this mammary gland has got a number of lobes, 5 to 12. The number is now. This is about the complete part of it. Okay. Now, then after this, you have to go to the mother's feed, and this is one of the basics. If you remember PCM, protein calorie malnutrition, marasmus and kwashiorkor. Kwashiorkor means neglected child. One of the reasons for kwashiorkor is more weaning away from the mother's feed and substituting with another diet. So that will lead to PCM as the children grow. So the first the breastfeeding is necessary for the health of the normal baby. Not only for that, for the lifelong resistance will be more in there. Now this is about the complete reproductive system after parturition. In this one part we left out is only the mammary gland. Okay, the mammary gland, as you know, because when it came to the secretion, lactation period, the period the mother feeds the baby is known as the lactation period. Gradually, it varies from body to body, physical strength of the individual. In some people, the secretion will be more abundant. Some people, it depends on and during this feeding time, the baby is born. When your embryo at least 4, suppose the baby has gone into the protected embryo where inside you are nicely within a liquid, protected completely, like a AC room like. When you are thrown into outside, you will be first facing into all the infections and anything. The child will develop, or the mother has got any sickness or cold or fever, automatically when they feed, the child will get the cold. Now, in that case, I cannot give one newborn baby a medicine. They can, nowadays they do inject. But the best way to treat is, they will give the medicine to the mother. Suppose the child is showing some cold or cough or something, the newborn baby. Now they will tell the mother to take the medicine because all these chemicals will be passed on through the milk to the mother, to the baby. So it's another advantage of breastfeeding the child. So not only gives you immunity, you can administer any medicine which you want to the child instead of directly poking the kid, a newborn kid. And you can give that medicine via, it's like a channel through which it can be fed. So when you come to this lactation period, mammary gland, you know very well, the mammary gland is, it has got a number of like a mammary gland structure in a pectoral muscle, skin gland, modified skin gland. It has got the mammary gland. Okay, very important this word. It's a characteristic feature of mammal. The word mammal comes from the mammary gland. Now they are viviparous animals, they feed the young, that's why they are called mammals. And the mammary gland, it is a modified skin gland. Okay, and second, it has got glandular tissue with the number of areolar tissue in between, and the gland covered by the completely padded with the fat around it. So the glandular tissue is known as the mammary lobes. Now there are a few lobes are there sectioned. Now, in between, they say there are lobes over there. Now, in each, this is a mammary lobe. You have got the areolar tissue over here. Areolar tissue completely surrounded by the fat, padding up of fat. Fat tissues will be there. Now, this is anterior. Posteriorly, you have the pectoral muscle. Now, this diagram is there in your book already. If there is a diagrammatic question, they can give you the diagram. Label the part. Diagrammatic question comes, you have to be very thorough because diagrammatic question is the most trickiest one in the case of NEET because I can give the diagram. I can label the parts wrongly and ask you to find out the mislabeled one. Or I can label the unlabeled parts I can give you. Label 1, 2, 3 parts I label and 1 or 2. A and B will be labeled, C will not be labeled. I can give it in a multiple choice. Or I can give take a diagram, label it A, B and C without labeling anything, match the following. A, and fix up A to whatever, 1, 2, 3. So diagram question is there. Last 2 years, there's no diagram question, they say, but diagram question, you should be thorough with the diagram. If you don't know diagram, you cannot study biology. If you want to expertise yourself in biology, every little thing what you got in your head, you must have your own diagram. You should be able to, if I draw, the cell is bigger, you should be able to draw bigger. If you learn to draw diagram mentally as well as of your own, then you can master the biology. And particularly zoology, anything, whether it's a botany or zoo, without diagram, you cannot master the subjects. So whether they ask the question or not, if you want to learn the fact thoroughly, first learn the diagram. So you have the diagram there, pectoral muscles at the cut end of the ribs, so the ribs part will be there, the pectoral muscles will be shown here. Now these are fat, completely padded with fat. That gives a spongy appearance for this organ. Now this is the lobule, mammary lobes. The number of mammary lobes, 5 to 12 or 5 to 10, the number is given. Number is one of the objective question. How you have got here testicular lobule? In testis you have got testicular lobule. The number of the testicular lobule you should know. And each testicular lobule got 2 to 4 seminiferous tubule. How the lobules are formed by the tunica vascularis. Innermost lining layer is tunica vascularis. That divides the testis tissue into lobule. Here in this case, it's only area, the lobule, and there are areolar tissue and fat deposits. From each lobule, you have got secretory unit. That is called alveoli. Just like your alveoli in the lung, there are, and these are the secretory units. This is only lobe. This is where the milk, this is naturally the secretory units means what? Alveoli means a sac-like structure. It will be lined by the epithelial cells, which is secretory cells. So the milk secretion is here. Now each, now this is the lobule. Just to show the different parts. Okay. Now each lobule you have got, these are alveoli. Alveolar cells, these alveoli. From this alveoli, a number of small capillaries comes, ducts. These are the ducts from the alveoli. All the ducts form together, these ducts join together, form a lactiferous duct. Now the lactiferous ducts, these parts only important. Now these are capillaries. One second. Now this is the lobule, mammary lobes. Okay. Now from this you have got a number of alveoli. These are the alveoli filled with alveoli. From the alveoli, the first set of ducts. Now a number of alveolar ducts. These are ducts joined together. These are all the things united together, form a lactiferous duct. Then it enlarges into an ampulla. Now this is the ampulla opens into the lactiferous duct in the nipple. So this is the small, when you capillary tubes, tiny tubules from the alveoli joined to form the common duct. Then it forms an enlarged part ampulla that opens into the lactiferous duct. These are the three parts. That is the first part, the lobule, alveoli, tiny tubules from the alveoli joined together forms a single duct. The number of, so naturally there will be a number of things joining another duct and then opens into the ampulla. Now this ampullary region that continues in the lactiferous duct. So these are the major parts: lobule, alveoli, thin tubules from the common duct, ampulla, lactiferous duct. So these labeling will be these parts you should know mainly. Now the duct system, and lastly to open all the lactiferous ducts are in the nipple region of the breast part. That's all. Now this is how the structure of the mammary gland is given, but invariably the number of blue objective part of it, how does it open out? This lactiferous duct they'll ask you. This is called lactiferous duct. You can there in your textbook already this, if you have what will I say? If you are following a CBSE textbook, you can easily form this as a lactiferous duct. You have got lactiferous duct. This is the ampulla. ampulla, lobule, alveoli. Secretory units are called alveoli. Not much question, but they can ask you question. Mammary gland, because we have got quite a lot in the reproductive system. Now this ends with your human reproductive system. Next part which we have to go through. If you have any doubt, you can post it to them. And the next we will go to the reproductive health. Next chapter. If we proceed to the 12th standard. Okay, that's enough. Thank you. There I don't see any questions in this. So